Hello everyone, we're talking about the development of eye. In this section, we'll have an overview. So, in the overview, we'll first talk about the various sources of eye development as to what sources and how do they contribute to the development of eye. Then we'll move on to the formation of optic vesicle, and optic vesicle inducing the formation of the lens placode. Then we'll see what choroid fissure is and how do the hyloid vessels develop. So, first of all, in the sources of development, we have the neuroectoderm of the forebrain, which gives rise to the retina, the posterior layers of the iris, and the optic nerve. So, this is the retina, this is the optic nerve, and the posterior layers of the iris are actually formed by the neuroectoderm of the forebrain. Then we have the surface ectoderm of the head. Now the surface ectoderm of the head, it forms the lens of the eye and the corneal epithelium. So this is the lens of the eye and this is the cornea. So lens of the eye and the corneal epithelium are actually formed by the surface ectoderm of the head. Then we have the mesoderm, which lies between the neuroectoderm of the brain and surface ectoderm of the head. It forms the fibrous and the vascular coats of the eye. So the fibrous and the vascular coats of the eye are formed by the mesoderm, which lies between the neuroectoderm of the forebrain and the surface ectoderm of the head. And finally, we have the neural crest cells, which migrate into the mesenchyme from the neural crests and form the choroid, the sclera, and the corneal epithelium. So choroid, the sclera, and the corneal epithelium. So this is the cornea, this is the epithelium of the cornea. The choroid and the sclera are formed. This is the sclera here in white. So all these parts are formed by the neural crest cells, which migrate into the mesenchyme. So we have four different developmental sources. The first being the neuroectoderm of the forebrain, the second being the surface ectoderm of the head. Now, in between these lie the mesoderm, which is the third source, and the fourth source are the neural crest cells, which migrate into this mesoderm. So, the development of the optic vesicle and the lens placode, it begins at 22nd day of intrauterine life. Now, a pair of shallow grooves or sulcuses appear on either side of the forebrain. So, this is the developing cavity of the forebrain, and these are the pair of the of grooves or sulci which develop on either side of the forebrain. Now, they tend to enlarge, they invaginate the surrounding mesenchyme to form the optic vesicle. Now, these optic sulci, they enlarge and they tend to penetrate the mesenchyme which surrounds the optic vesicle. Here you can see they're now migrating in, invaginating the mesenchyme. So, at this point in time, this enlarged optic sulcus, it forms the optic vesicle and induces the formation of the lens placode. So optic vesicle, it grows laterally. Here you can see it is growing laterally and it is expanding and it is increasing in size. The proximal part is connected to the forebrain. Here you can see this is the proximal part which is connected to the developing forebrain. It constricts to form the optic stalk. So there's constriction in this portion to form the optic stalk. And now the distal part, it comes to lie in contact with the surface ectoderm. So this in blue is the surface ectoderm. Now it comes into contact with the surface ectoderm, and at the point of its contact of the surface ectoderm, there is thickening and there's formation of the lens placode. So initially an optic sulcus was formed which moved laterally, enlarged, and induced the formation of lens placode in the surface ectoderm. So this is the entirety of it. Recapping it again, first of all, optic sulci appear as paired growths on each side of the developing forebrain. These optic sulci, they invaginate the surrounding mesenchyme. They move laterally and enlarge in size. Now the proximal part which remains in contact with the developing forebrain, it narrows or constricts to form the optic stalk. The distal part comes to lie in contact with the surface ectoderm and induces the formation of the lens placode. Now moving on, We have, this is how the optic vesicle develops. Again, this is another view. This is the cavity of the forebrain. This is the optic sulcus being formed. The optic sulcus now increasing in its size and moving laterally. As it is moving laterally, it is invaginating the surrounding mesenchyme and inducing the surface ectoderm to form the optic lens placode. At this point in time, the communication with the forebrain cavity, it forms the optic stalk, and the distal portion of this optic sulcus forms the dilated part, which is the optic vesicle. Now, let's talk about the formation of the lens vesicle and the optic cup. So now the lens placode shown in blue in the figure, it gets depressed below below the surface to form the lens pit. So here you can see this is the depression in the surface. This is the optical vesicle, and this is the lens placode. So this lens placode, it undergoes a depression below the surface, and it forms a lens pit here, as you can see in the figure. This lens pit, it tends to deepen. It deepens its edges and approaches each other in the midline. Here you can see it is deepening and its edges are now approaching each other, and now these approaching edges, they fuse to form a spherical lens. Here you can see the edges have now fused and formed a spherical lens. Now they're cut off from the surface ectoderm. Now the lens has been cut off from the surface ectoderm, and the optic vesicle, this is the optic vesicle, invaginates and forms a double layer optic cup. Now it invaginates on itself and forms a double layer optic cup. So the lens placode, it undergoes indentation. The indentation moves downward and inward. This indentation then folds upon itself to form a circular lens, which is cut off from the surface ectoderm. Now the optic vesicle, which was initially inducing the formation of the lens placode, it folds upon itself and forms the optic cup. So let's talk about the choroid fissure and the hyloid vessel development. So choroid fissure develops on the inferior aspect of the optic cup and the optic stalk. So this in yellow is the optic cup, this is the optic stalk. Optic stalk is the communication with the developing forebrain. Optic cup is the distal dilated portion of that optic vesicle that was formed. Now choroidal fissure, this is the choroidal fissure in gray. It develops on the inferior aspect of optic cup and stalk. Now the vascular mesoderm enters into the choroidal fissure. So vascular mesoderm finds its entry into the choroidal fissure, which is shown in gray in this figure. So it gets trapped in the optic vesicle and the optic stalk. So this vascular mesoderm, it enters there and gets trapped into the optic vesicle and stalk. It forms the hyloid vessels. These are the hyloid vessels. Here you can see. So these hyloid vessels are formed by the mesenchyme that actually gets trapped in the choroid fissure, which is formed in the inferior aspect of the optic cup and the optic stalk. Now the distal part of these vessels degenerates, while the proximal part persists as the central artery and veins of the retina. So the distal part, it degenerates. However, the proximal part, it stays, and it stays as the central artery and vein of the retina. Here in the figure you can see this is the optic cup, this is the optic stalk, this is the choroid fissure which has been formed. So this choroid fissure is penetrated by the mesenchyme, which stays there and then gives rise to hyloid artery and hyloid vein. The distal part of this hyloid artery and vein, it degenerates. However, the proximal part persists and forms the central artery and vein of the retina. So this was all about this section. In this section, we saw how the development of eye begins and what are the various steps that are taken in development of eye. We saw how the hyloid vessels are formed. We saw how the optic cup is formed, how lens placode is formed, how lens in itself has been formed. It is important to remember the sequences as they're happening continuously in a certain fashion. I hope you understood it. For further such sections, keep watching sqadia.com.